If you can't catch it at stage one, your survival rates for almost, or your cure rates for almost every cancer drop dramatically. And the reason why is, of course, is that tumors grow 5 times faster in dogs than they do in people. So there's a much narrower window that you can catch that cancer and actually cure it.
Welcome to the Canine Cancer Alliance webinar. We try to do these webinars regularly, roughly once a month, and try to cover some of the latest research findings and latest discoveries and anything that we can do to help our dogs fight cancer. This picture is Gus. He was diagnosed with osteosarcoma. We saw a little bit of limp one day. The vet x-rayed him and said it might be osteosarcoma, and we were terrified. But we thought maybe we caught it early. But unfortunately, by the time osteosarcoma shows symptoms, it's already starting to metastasize. You can't see it in the lungs, but micrometastasis is taking place. And so we are very interested in early detection as well as therapeutics that could potentially lead to a cure. For those of you who don't know what Canine Cancer Alliance does, we're a nonprofit foundation founded about 9 years ago, and our mission is to end canine cancer as we know it today by supporting and funding research. So we are also stressing education, and so that you can learn about different clinical trials, not just the ones that we're supporting. You may have joined us for a webinar last October called What Causes Hemangiosarcoma. This was led by Duke University researchers who has launched a new study funded by our donors that is looking at the correlation between environmental toxins and hemangiosarcoma occurrence in golden retrievers. They are already seeing that dogs who are exposed to more environmental toxins are likely to end up with bladder cancer. That was a bladder cancer targeted study. But this study is going to be focused on hemangiosarcoma. In human cancer, they know that there are now earlier occurrence of cancer, especially colorectal cancer in younger people, and that is correlated with life factors and other genetics. It would not be surprising if we find out that for dogs, similar thing is happening, that we are all being exposed to more environmental toxins, and that might be exacerbating cancer. If you go to our website and look under /webinar, you'll see various webinar recordings, including the one about hemangiosarcoma and environmental toxins. We did a couple of liquid biopsy and cancer mutation analysis webinars last year. Dr. Guanan Wang from Vetomics, she has a new test that looks at the mutations in cancer tumor samples. So that test is available for families and vets so that you can find out if there might be better personalized or targeted therapy drugs that might help your dog. Another webinar about 2 months ago was done by CEO of CanCan Diagnostics, and they have liquid biopsy that looks at cell-free DNA, and that test may also help disease monitoring. So instead of waiting in the dark and waiting for an CT scan or x-ray to see if cancer has come back, blood tests might be used to see if the treatment is working and how cancer might be progressing. And last month we had Dr. Mark Mamula give us an update on the canine cancer vaccine study that he is leading, and which is enabled by your donations. If you're interested in finding out how to get to that trial and learning more about it, please watch that webinar or contact us. I'm going to mention that all this is possible because of donations. We have something called Wag Love Life, Walk and Run. And for people who live in Washington State, please join us in Redmond, Washington. People can walk virtually. So if you're interested in creating your group locally, please let us know. Today's webinar on the topic of early canine cancer detection is led by Dr. Stephen Johnston. Dr. Johnston has been doing research for decades at Arizona State University on sort of the intersection of biology and semiconductor technology. And he has come up with really a number of innovative ideas. And he started a company called Calviri, to commercialize many of these ideas. So today he's going to be sharing with us what he's learning about early cancer detection through his trials and a new test that is becoming available from Calviri. And also, I hope you'll have a chance to describe what else he's doing at his company in terms of cancer prevention and therapeutics. So, with that, I will hand over to Stephen.
Okay, good. Well, thank you for having me, Mara. Your association's doing a great job, and I really applaud your efforts. I knew of Mara back from when she was in DARPA, which was ironically traced back to the origins of one of the technologies I'll tell you about. I worked with DARPA for many years. I'll just start right away with getting into the information, and then this will be relatively short, but I'll be glad to answer any questions you can. Mara, if you want to interrupt me along the way, that's fine also. So I started this company with my co-founders in 2018, and we decided that I actually had decided many years before, I'm an inventor, that I'd figure out how to invent something to end cancer. It seemed like a good thing to do. And the motivation for that, I don't have to tell you about this, but most people don't realize that for humans, there's 20 million cases a year in the world and 10 million deaths, but 70% of those occur in developing countries. So any solution to end cancer has to be an inexpensive solution, which is not the mantra for modern Western oncology. So I think we actually laid the groundwork for accomplishing this. And there's two things that we worked on. This one I'll tell you the most about. This is our efforts to develop a universal diagnostic. Not quite a tricorder, but getting pretty close to it. So to do that, we had to, as Mara mentioned, we had to take a totally different view of how to approach diagnostics than everybody else had been doing and still are. And so to do that, we merged the semiconductor industry with peptide synthesis. So we built a machine that looks like it makes computer chips, but it doesn't. It takes wafers and it makes little squares, as you can see here. And each one of those we use photolithography to make peptides, to synthesize peptides. And this is one of them, and you can see the little squares on there that we have. So each one of those is a diagnostic. And on each one of those little squares, we apply 5 microliters of blood and wash it off and look at the pattern of antibody binding. Amazingly, this is a universal diagnostic. As far as we can tell that we've tested so far, it works for any species and it works for any disease. And it's quite simple to do. We also, I'm not going to talk about this, we wanted to also do something better than even detect cancer, and that is to prevent it in the first place. So the way we figured out to do that is that the best place to do that would be have a vaccine that could prevent it, just like infectious disease. And so to do that, you have to have some foreign parts of a tumor that are in common across all tumors. Otherwise, you couldn't make a vaccine to prevent it. And what we discovered is that tumors not only make mistakes in the DNA, as everybody knows about. I think you had a webinar or something on this, or a newsletter about personal cancer vaccines. But what we discovered is that they make 100 times more errors in processing their RNA. And those errors create what are called neoantigens, little bits of proteins that look foreign. And those little bits of foreign proteins elicit antibodies almost as soon as the tumor starts developing. But what was important, we discovered that those little foreign bits are shared across tumors and across patients. So that allowed us, if we could screen those, to develop a vaccine that could potentially prevent cancer. And so we've created the infrastructure to do that. We're pretty confident now we can make a vaccine for dogs and for people, that it'll be slightly different components that could largely reduce the incidence of cancer. And eventually we'll be testing those. But even though we started as a human company, we could get no interest in developing these products in the human side for a number of reasons. And so we decided to turn to dogs to do that first, both as a validation process that could be done faster in terms of approval process, faster to revenue and validation of that this works, and get to humans through dogs faster. This may not be as important to you, but as important to investors and to our company is that people underestimate the market potential of dog oncology. And this is essentially a totally untapped market. The largest untapped market in dogs right now is dog oncology. They have very large market potentials. So,
Can I just interrupt you? I see that number often,
which
and
number?
the size of the market.
Yeah.
And so when I see these market potential, I cringe. We want investors to be interested, but we want a therapeutic or diagnostic to be very widely available to help as many dogs as possible with reasonable profit, but not just like really maximizing profit. So it's going to be $40,000, $50,000 therapy.
So that's an important point, because if you recall what I first said is that if you want to address cancer worldwide for people, it has to be inexpensive. And that's the same thing true as dogs. So we approach this like an engineer. Okay, what are the specifications that you have to have for this product? One of the specifications is that they all have to be priced at under $100. So that was this TAM, as they call this, total addressable market, is based on a product that's priced at $100. And then using the number of dogs that are over 5 years old, which is in the United States about 47 million dogs, as those would be the screenable dogs. And that's and once a year. So that was the specifics for arriving at that number. What will really determine it will be the adoption rate, right? So if it's priced at, like PetDX had, at $500 to $1,000 to test your dog, and they couldn't even pick up stage one, well, it's not going to get a good adoption rate, as PetDX failed. And so those are the realities behind these TAMs, and we're well aware of those. The same thing through the preventative vaccine. That's the same sort of idea. Price at $100. All dogs over 5 get this vaccine every 2 years. That was the specifications for that.
That's great to know. Thank you.
Is that okay? Okay. But let's focus on the diagnostics. And on this figure, there's a couple interesting numbers. One, there's about 6 million dogs a year that are diagnosed with cancer in the United States. What's remarkable about that, that's 3 times the number of people that are diagnosed with cancer. And so that's a big number, right? What you may not realize is that, and in fact, Mara, you just gave us an example of that, only 4% of the dog diagnoses are during their wellness exams for cancer. 88% of those diagnoses occur that owners, just like you did, notice something in their dog and their behavior, go in, and it gets diagnosed. What that means is almost always those diagnoses will be at stage 2 or later. And stage 2 or later is not good. It's much worse than in humans. Whereas, for instance, in breast cancer, stage 1 and stage 2 in humans have almost the same 5-year survival rate. They're very high. In dogs, that is completely different. If you can't catch it at stage 1, your survival rates your cure rates for almost every cancer drop dramatically. And the reason why is, of course, is that tumors grow 5 times faster in dogs than they do in people. So there's a much narrower window that you can catch that cancer and actually cure it. And that's the reason we focus completely on being able to have a product that could diagnose at stage 1. You'll hear other people go out and say, I can catch early cancer with their diagnostic. Ask them what their sensitivity for stage 1 tumors are, and you will be surprised, very surprised at how low those numbers are, or that they don't even have those numbers. And the reason why is that and this was a big problem for us, we said, okay, if we're going to have any impact, I'm not going to make a product that's another Me Too that goes out there and says early and really means late. If we can't pick up stage 1, we're not going to have a product. But in order to develop that kind of product, we had to have serum samples from dogs with stage 1 tumors. No one had any such collection. So we spent 4 years collecting samples all around the country from any dog that we could that was diagnosed with stage 1 tumors, qualified clinical assessment, so we knew what they were, and then bring that into our sample base. So we're the only people in the world with a large collection of serum samples from stage 1 tumors. And that was absolutely required to do what I'll show you. And in fact, our controls, we took dogs, got samples from dogs that were tumor clear, and then followed them for 2 years to make sure they really were clear from tumors, that they didn't have a latent tumor at the time of our sera draw. So we went to a lot of work in order to be able to develop this product.
How many dogs?
About 700 with tumors. Yeah. So this is the problem here. Your wellness visits don't do you much good in terms of cancer diagnosis. They may do it for other metabolic diseases and things like this, but not cancer. So we set out to develop a diagnostic that could pick up at least the 5 major cancers at stage 1. And so we made that collection, and this is an example of putting these dog sera on our peptide arrays, and each dot represents a positive peptide. So these are the cancer types: soft tissue sarcoma, osteosarcoma, mast cell tumors, lymphoma, and hemangiosarcoma. Those account for about 70 to 80% of most major cancers in dogs. So this is actually what we saw on these arrays, is that dogs that have HSA mostly had these sorts of peptides reactive. Dogs that have lymphomas had these. Dogs that have MCTs, et cetera, et cetera. And these signatures of peptides on our arrays is what actually composes the diagnostic. There's something quite remarkable that we have seen that we haven't been able to follow up. There's a set of peptides that occur in dogs without tumors that we've never seen any dog with tumor have. So there's a signature of not having cancer. We don't know what that means. We haven't had the bandwidth to follow up, but it's something we will in the future. So we think with this kind of diagnostic that we developed, that we call stage 1 plus, that's trademarked now, that we'll be able to switch. This is the actual results when we end up screening all those dogs, is that this is the statistics that we got for the performance of the tests that we had on the 700 samples that we had. The sensitivities ranges from 60%, holding the specificity high. So at these specificities, we say sensitivities from 60 to 88%. The other tests that are out there, let me give you an example, mostly from humans, because there is no good data from dog, the PetDX or the existing ones that are out there for stage 1. But for humans, GRAIL has a sensitivity of 18% for stage 1 breast cancer. So that's the problem with these nucleic acid-based tests. They can't pick up early tumors. So we think we can improve on this, but this is dramatically better than anything that's ever been reported before. And as far as we know, this is the only test that has shown high sensitivity for stage 1 tumors. So with that test, we think we can change this profile from you going in for your wellness exams, from only picking up to 40% to maybe picking up to 90% of the tumors at a wellness exam. And most of those will not be stage 2 to 4, but most of those will be stage 1. So that was what we're trying to dramatically change about the profile for cancers. And we think that will have a big effect, because where the studies have been done, where they compared the cure rates for stage 1 versus stage 2 or later, you can see, here's what you see currently in the tests that have been reported for stage 2 or later. If you look for stage 1, there's much better survival rates and cure rates for discovering cancers at stage 1. For some of them, like anal sac and hemangiosarcoma, the difference is dramatic, huge amount. So
Can you just explain what you're plotting here?
So this is the 2-year survival rate, which is basically a cure rate for dogs. And so if your dog is discovered at stage 2, in this example for hemangiosarcoma, there was almost no survival dogs for getting care for a stage 2 or later. All of them were stage 2. However, if your dog gets the same care and it gets as detected at stage 1, the survival rate, the cure rate is about 50%. So these are the only published papers where they've compared the survival cure rates between stage 1 and stage 2. We think they'll apply to almost every cancer that's around. Is this clear to people?
I think so, but I'm still a little bit surprised with your hemangial example.
Yeah.
So the blue one is 2-year survival rate if it's caught
If
at
discovered at stage 1.
stage 1, and the purple is
2-year survival rate if discovered at stage 2.
so with hemangiosarcoma, if it's stage 1, it's localized. So
Yep.
splenic hemangiosarcoma, if it's localized, and then you have a chance to get splenectomy, right? And
Right.
then and you say, okay, hopefully it's gone. And then you do chemo, but
Yep.
chemo doesn't really make that much difference. It does help extend the survival time, but I'm still surprised that if you find it at stage 1 that you get a 2-year survival time of 50%. I think there's something a little bit funny about
No, this
that
is
data.
published data.
Which paper is that, by the way?
This is
Yeah.
by David Bales and crew that was done. Actually, this is old. It was published quite a while ago.
Okay.
There aren't that many of these studies that have been done
Yeah.
that both sets of dogs received the same. Yeah, this is published later. If you don't believe it, Mari, just look up the paper and you will see
I
the
will.
result.
I will.
Okay.
No, 2-year 50% survival rate for stage 1 hemangiosarcoma. Yeah, I
But
will
why you
definitely
don't,
look that up.
you know why you can't, because dogs are almost never discovered at stage 1 in hemangiosarcoma. We are conducting a trial right now that's a therapeutic trial at 3 sites, Colorado, Wisconsin, and Davis. And we started this over a year and a half ago. We've developed a vaccine, off-the-shelf vaccine for hemangiosarcoma. So the trial is to have dogs discovered with stage 1, localized tumor, only for splenectomy. We don't include heart in this. And so only splenic tumor, we treat it with either standard of care and our vaccine. It has taken us over a year and a half at those 3 sites and around those sites to get 53 dogs.
It's hard to get stage 1.
Yeah, so that your impression of stage
Yeah.
1 is because there's almost never discovered at stage 1 tumors. Yeah, because that's if you went to heart or sub-Q, it would even take longer to enroll enough dogs.
Yeah.
Only about 10% are heart, for instance.
Yeah.
Okay, so we're pretty confident our diagnostic can convert the dog cancer protocol from looking like the purple in yours for very low survival rate to much better survival rate. In some cases, very high survival rates and cure rates effectively in a dog. So that's what the goal is. This is what we'll be coming out with. This is what it actually will look like. This is what you'll go to your wellness visit. We'll take a small amount of blood at the same time that you're sending off to IDEXX. We'll send a small amount to us at our reference lab. That drop of blood is applied to one of these chips. This is our pattern. This is a small part of the array where each one of these little squares is a peptide, and the amount of intensity of binding is the amount of antibody bound to that particular peptide. So as I said, our data has already been published. The data on all of the survival rate, that's all published, so you can look it up. And if you go to this paper, you'll find all the data on the survival rates also. So this is what we want to come out with. We'll come out with it in about a month or so on a small scale, not on nationwide. But what we will be able to do is detect all stages at stage 1, because we collected them from 5 major tumors, but we also collected any other stage 1 tumor that occurred. So we have rare cancers that are also in that 700 that we screened, some brain cancers, et cetera, prostate cancer, things that you almost never see in dogs, but they were included. And so we can pick up whether it's cancer or not cancer, we can pick them up with high sensitivity. Now, I saw you mentioned that people were interested in how accurate could we predict what kind of cancer the dog is. So we can, for those 5 cancers, the major cancers I listed, we tell you what kind of cancer with a specificity between about 60 and 99%, depending on which one. The worst by far is osteosarcoma. That is, we have relatively little specificity for osteosarcoma. The other cancers were fairly accurate in saying what type of cancer it is, but at the time of the release of the test, our confidence limits on those predictions are fairly large. So that we're not highly confident about designating the type of cancer it is. And that was because we only had between 30 and 60 samples from each individual type of major cancer. But as we launch the product and we discover more stage ones, then we will be able to increase that number and then increase the specificity of the test. Because
Stephen, can you talk a little bit more about why specificity is so important, like in terms of false positives, or just so that people understand better?
Yeah. So in the human space, that's been a big point of contention is that just a little history, when people started talking about doing liquid biopsies for assaying for cancer, the biggest concern was the specificity. That's how many times did you call a cancer and you're right or wrong. So if you're 99% specificity, it means 1% of the time you tell somebody they have cancer and they don't. And so the concern was, what about that 1% that you're wrong about? Will they have treatment that they shouldn't have had? Would they go to expenses that they wouldn't have gone to? Will they have anxiety that they wouldn't have had otherwise because you told them they're positives? Ironically, it has turned out the nucleic acid-based tests have very high specificity. If Grail's test tells you, Galleri tells you that you have cancer, you have a 99% sure that you do have cancer. Now, they may only pick it up at 18% of the time if you do have cancer at stage one, but if they tell you do have cancer, you do have cancer. So the same thing is true for dogs. So if we pegged our specificity at 87, 97. So that at that specificity where we'll make 2.5% of the time, we'll be wrong. We'll say you have cancer, but you don't really have cancer. Your dog doesn't have cancer. So and the concern there is, okay, what would a dog owner do at that point? First of all, they'll have confirmatory diagnosis. So if we said that we think you have HSA and we predict that, you would go in for an ultrasound for that before you would elicit any treatment for a splenectomy or chemo. So that would be the implication for the dog owner and the vet.
If you chose, say, 99% specificity, so lower false positive rate, like 1%,
yeah,
then your sensitivity would
drop down.
Okay. Yeah.
So I actually think it's the reason why we stressed the sensitivity is because if you don't catch a cancer in a dog it's bad. It's really over with if you don't catch it with stage one. So we focus more on the sensitivity than we would have, let's say, potentially in humans because of this compression of the time scale for the growth of the tumor. But what we do as a product, what we will do is that if we say that you have a tumor for your dog, we will offer to test that dog again for free in 2 months. So if the tumor is growing, then the antibody levels actually start going up. And then you're more confident that really your dog really has a tumor. If your dog in humans, particularly in breast and lung cancer, about 25% of those that are identified early just stay latent or even go away. So the question is, would that happen in dogs? No one knows if that happens to dogs. Not enough dogs have been discovered in stage one to know what frequency of the time do they stay latent or even go away. But we will retest, and if the tumor is growing, the antibody levels will go up, and then you definitely want to do something about that in your dog.
See, so you're not just measuring, you don't just have a threshold and measure the presence of antibody, but you're actually measuring the concentration or
Yeah, we
number
have a
of antibodies.
yeah, and Mari, I'm sure you'll appreciate if you have the first signature and you're comparing it to the second, you have much more resolution because you already have a baseline for that dog.
Okay. So this test could be used then for monitoring disease.
Oh, yeah, yes. In fact, what this test is not good for is for minimal residual disease. The nucleic acid tests are very good once you've been diagnosed with a tumor and you or your dog, and let's say they excise it with surgery. The nucleic acids in the blood have a half-life of about 30 minutes, so that if your tumor is really gone, all the nucleic acids for that tumor should disappear.
So this is a cell-free DNA with all the cancer mutations. Those
Yes.
DNA in the blood disappear. But in your case,
Yes.
you're not looking at DNA, you're looking at antibodies.
Yeah, so those antibodies have a half-life about 21 days. So they're not good for monitoring, did I get all the tumor or not? What they are good for, though, is reoccurrence. So our first product for people will be for testing for the reoccurrence of cancer. You've already had cancer, you'll test it every 6 months to make sure that it's detected early before it comes back again. So the same thing could be done in dogs. But it could be done, and people, I think, will apply this same test for reoccurrence. But our focus in terms of getting people to use it is to get that stage 1 in the first place.
Okay, that's awesome. Thank you.
And then there's 2 other leading tests out there that are being advertised, I think, falsely as saying early detection of cancer. Grail does the same thing, Garden and Exact Science. They all say early detection. But if you look at the actual data, they perform very poorly at sensitivity for stage 1s. And so I think on every category, our test will always pick up later stage also, but that's very easy. By the time it's late stage, there's a huge amount of antibodies being generated and stuff like this. Interestingly, in people is where we've done the most tests. At stage 4, the immune system basically gives up. It just collapses. All the signature for that the tumor was the patient was battling against the tumor just gives up on it. And so it's really interesting.
And so we're going to go through the questions later, but I just wanted to bring up one question here from Donna. She says, I have a dog that was diagnosed with hemangiosarcoma almost 2 years ago. The spleen was removed. Tumor on the liver has gone away. Will this blood test help me determine if she still has cancer? Currently doing holistic with her.
Yeah, it can potentially if there's still HSA in the dog system, and it could well be that there was metastasis that was in there, and the dog's immune system is basically keeping it in check, and that's good. If that dog was monitored, we'd recommend monitoring that dog every 6 months at least to see if there's any indication that signature is increasing, which would mean that the tumors are starting to grow again. Okay. Yeah, so I think there's a lot of attractive features about this. This is a very small amount of serum. We can detect all cancers, whether you have cancers. But for most cancers, we can give an indication of the type of cancer. And as we expand the test, it will get better. The more numbers we get, we'll get better specificity, sensitivity, and we'll be able to be more accurate about telling you, even for rarer cancers, what kind of cancer it is. Tests that out there are limited in terms of lymphoma and hemangiosarcoma. They only do later stage.
I just want to share my personal experience with the tests.
Before we continue, Canine Cancer Alliance is a nonprofit dedicated to accelerating research for safer, more effective treatments for dogs with cancer. We fund promising studies, support clinical trials, and share science-based insights with a community that believes dogs deserve better options. If you'd like to learn how you can be part of this effort, visit ccralliance.org.
My dog, my golden retriever, is 9 years old, was just acting very lethargic for months. So I knew something was wrong with her. So finally, our vet did lymphoma test. IDEX has a lymphoma-specific test.
Yep.
And came back positive. Now, this is a family vet, and she said, according to IDEX, specificity is very high. False positive rate is so low. There has to be lymphoma. So we had all sorts of tests done to see if it was lymphoma, and it wasn't.
Yeah, and I hear these stories all. I hear a story for that's the IDEX test. I can tell you for the other tests. I had we were just talking to a vet out here that's enrolling our test, and she said, "Oh, I gave this test to my dog, and it was clear." And then a month later, she had late stage hemangiosarcoma, and that was supposed to be covered by the test. And so I hear these stories all the time. Now, no test is perfect, right? You know that you can't expect that. But I think that one of the things is that as it gets later, it starts to get a little harder to tell what type of cancer it is because the signals start to merge over. It's a little easier at early stage to tell what's what. So I can understand why there might be some misclassification as a type of the cancer. I would say though, Mark, at least they discovered that they had cancer, even if it wasn't a lymphoma. So it's not all bad news.
Yeah, that's very true.
Yeah, and the lymphoma ones are particularly hard to do for early stage. And I can tell you that at least one of these companies did not even test for early stage. They had no samples to test because they actually purchased them from us.
So just for the audience here, we're doing a community-based study, tests that are marketed directly to families. If you'd like to share that experience, please email us.
Very interesting. Yeah, no. And in fact, for us too, we would like to get that feedback because we think our tests can be improved and we like to get that sort of information back. Okay. This is our test. It will be coming up. We're starting it locally and in Southern California mostly with private clinics that will be launching it. So it will be priced at $100 to your vet. What the vet wants to do from there is up to them, but we will let you know that's what they're paying for it. You can negotiate with them about what they charge. Let's see. These are the capabilities that I talked to you about. Even though our name is stage 1, we can do later stage. It's just that's not where the emphasis should be. I won't go into this but this is our company. And by the way, just so you know, I'm not just blowing smoke in terms of these inventions. I'm pretty good at inventing things that are only in class. John Sanford and I actually invented the concept of using nucleic acids, RNA, and DNA as vaccines. We invented that almost 30 years ago. So it took a while to get recognized, but it was in your COVID vaccine. So we've had a pretty good track record of inventing things that everybody said couldn't work, and they did. It just took a long time to get recognized. Hopefully these won't take so long. So I think that's it. I'd be glad to get other questions that people have or comments.
Okay. And how's preventive vaccine working? Has that trial ended?
It ended and it was extended. We're just finishing up the study of its effectiveness. The key thing is the immunology that we've had to do on the dogs to see how many dogs had vaccine takes and what peptides they responded to and things like that. And so as soon as I can, we'll finish that and report that out. But we know we can make a much better vaccine now. We've shown that technically what happened when we got the money to do that test, we only had dogs with late stage tumors available to screen to figure out what to put into the vaccine. We later discovered that dogs with late stage tumors have totally different neoantigens than early stage. And so the actual vaccine that we'll take into the USDA will have a completely different composition than the one we tested before. And we do have as I said, we have a therapeutic vaccine that's being tested for hemangiosarcoma that goes on for about another year.
So if the late stage antigen profile looks very different from early stage,
yeah,
I'm just wondering if maybe this combined with something like palliative radiation therapy, something that induces in situ vaccination of just releasing tumor antigen directly in the dog's body, does that make more sense?
You mean to combine it with our vaccine?
Yeah.
Yeah. I think that's probably true for late stage. And I saw on your website, you actually posted these results, I think, is that what's clear on the human side, that the personal cancer vaccines only work in combination with Keytruda. They do not work by themselves.
The immune checkpoint inhibitor, like
Yeah.
Gilvatmab for
So
dogs.
they have to have
Right.
in this, it'll be
Yeah.
Gilvatmab for dogs. So we're
Yeah.
exploring, when we get the bandwidth, we'll explore the possibility of combining our off-the-shelf therapeutic. We'll have a therapeutic vaccine that's off the shelf for any dog cancer. We'll explore, if we can, combining Gilvatmab with our therapeutic vaccine, which may be necessary even for stage 1 tumors. It's so obvious is that if you're going to do something for cancer, you want to do it before it occurs, not after. And you think would be obvious to everybody, but it apparently is not. So it's much easier to prevent cancer than it is to treat it.
Someone is asking about non-splenic hemangio, so cardiac, subcutaneous, or other visceral.
Yeah, so that's an interesting point. We haven't had enough of them to tell if there's a signature difference between cardiac and splenic hemangiosarcomas, for instance. We can tell B from T cell lymphomas. That looks like that's doable. Although they have a lot of overlap in their signatures, they're probably distinguishable. But like for all of this, at least because our focus is on stage 1, so we haven't tried to look at those. It's much easier to get samples at late stage to ask those kinds of questions, but they're not useful in terms of really helping the dog. So we've focused on stage 1, and we just haven't seen enough cardiac tumors in order to determine whether we can distinguish those.
Okay, good. Let's see who on the chat. Inflammation. Can inflammation or other confounding non-cancer condition affect the accuracy of your result?
I can actually check. That's my question. I just want to add one thing. My dog is hypothyroid and does have elevated her ALT liver levels. So I wasn't sure I shared that with him, but I don't know if that has anything to do with the inflammation. They thought it might.
Yeah, it may. But here's, by the way, I'm an inventor, not an oncologist. So take everything with a grain of salt. But early on, this was one of the biggest questions people had is how do you know you're not just measuring inflammation? Because these are antibodies. When you have an infection, you have inflammation, you get antibodies. So we actually directly tested that, and this is published, where we took 5 inflammatory diseases of the pancreas, pancreatitis, diabetes, pancreatic cancer, and asked, could we distinguish those from each other using this kind of chip format? And we could completely. And that's partly because what we're measuring on here are IgGs. These are antibodies that are developed with high specificity to whatever that offending molecule was, whether it's from a tumor or an infectious disease agent. So these are not the innate inflammatory response antigens. These are highly specific antigens, like your infections.
Thank you.
That's actually a really core question, right?
Yeah.
Okay.
Oh, and good luck with your dog. Sorry to hear that.
Thank you. We're actually, Marty, I'll reach out to you too. We actually have been in touch with the Can Diagnostics. We are going to do that test, and I'd be willing to do this test if it's available at our vet or we can somehow access it, but curious.
I don't think our test would be very useful for in your case, because ours is most valuable for picking up before it's diagnosed. Once it is diagnosed, you can monitor that with the nucleic acid-based tests.
Sorry, I had 2 shepherds. One had hemangiosarcoma and passed away. My current one has not been diagnosed with any cancer yet, but I'm curious because she's a shepherd and.
Yeah, no, I would but she says you already have symptoms.
Not necessarily. It just said it came back. This is just a study that they did. It wasn't like anything that's official, but they were just doing a study about it, and they offered to test her for it, but they said they've been having issues with inflammation showing up as cancer markers. I can
Okay, yeah, that I know which test you're talking about. That is a problem with that if it's the test I'm thinking of, because they're measuring nucleosomes, and nucleosomes are affected by a lot of things, even diet. This is if you eat, your nucleosome levels go up, and that may be an issue there. It is for late stage, yes. They have no data for stage 1.
You emphasized it's 5 cancer types, but then you also mentioned that because you have data, would you actually name the cancer type that they might have?
When it initially comes out, we'll have to make a decision on that because we're retesting this. There's 2 tests that we are completing. One is that we just did a field trial where we took dogs that were 7 years or older, just from multiple sites, that had no history of cancer and had a clear wellness visit. We collected their samples, and now we're testing to see how many of those we actually pick up that have stage 1. And so that's a real life put up or shut up kind of test. And so far the results look very good, but we will report that out. And then we're still debating whether to actually report for those 5 cancers on the what specific type it is, and are our confidence limits high enough or not. We may come out just saying it'll be cancer or no cancer.
Okay. And how many milliliters of blood is going to be sent?
Yeah, we're asking for 0.5 ml, but as I told you, the test only uses 5 microliters, but we don't want people sending us 5 microliters of blood.
Right.
It dries out too fast.
Does it have to be preserved? Is it a
No.
special kit or?
No, that's the other thing in your specifications. You don't want to disrupt the normal procedure that's already there. So all they do is use standard red caps, put that in there, spin it, and send it to us, just like they do anyway. We had to adapt it to the vet techs because they're the ones that are actually collecting this blood.
Okay. And what's the turnaround time for getting results?
Yeah, so we're promising it within 5 days, but I think we can get that down, but we don't want to go too quickly initially.
Is the test being done at your lab or distributed at one of these?
No, it'll be done at a central facility that we have set up. Eventually, it could be distributed, but just initially, we want to make sure it's done right.
Okay. So some of these tests that are consumer targeted, the test results comes back low, middle, high. Doesn't give you a number, but what does your results say?
We will give you a probability number for what, our estimate is that your dog has cancer, and here are the characteristics. And there are some dogs that are intermediate, and we will say there are no call. So it'll be cancer or no call.
Cancer or no call. It's not going to say osteosarcoma this percentage, hemangiosarcoma this percentage.
No, we don't know. We may do that. We just have to look at those numbers more carefully with some more samples. Are we confident enough? If we're not confident enough about the type of cancer call, we don't want to bias the results on that.
And what are the vets going to do then if it comes back as higher percentage?
The vets will then make an evaluation of what to do for that in standard procedure. Most of the vets will do if it hasn't been to a wellness exam recently, they'll redo a more extensive exam. We've already seen this happen where they do that and they pick up anal cancer because they forgot to do the screen. Or they pick up an MCT because it was much smaller than normal, but they just didn't catch it the first time. So a standard exam may pick this up again, or a lymph node that was swollen that they didn't detect before. And then they would proceed on that basis. If nothing shows up from that, they may look at the probabilistic. If you are a golden retriever, you have a good chance of this being hemangiosarcoma, so they probably give you an ultrasound at that.
Okay. Here's more questions are in the chat. Let's see. Well, you're launching on a small scale initially in Southern California. Could the vet clinic still procure the test in Massachusetts?
Yes, we have had specific people ask us to set it up in North Carolina, even in England and things like this. If there's enough volume there, we will, because it's the shipping costs. If you only have one sample and you're shipping from Boston, it adds considerable cost to it. But if there's 10 samples shipping from Boston from that clinic, we're glad to sign it up.
If the family is willing to pay for shipping, would you consider
Yeah,
one-off?
of course. Yes.
Okay. So it's just a matter of talking. Families can't directly contact you. They need to go through the vet. Vet has to agree, and then they'll reach out
They
to
can
you.
actually they can contact us if they have a vet and they refer us their contact. We will contact them and provide them the information about the test, and then they can offer it to the client if they want to.
Okay. So go to Calviri website
Yep.
and then look for the contact and for email address. Okay.
Eventually, this could be a direct to consumer because we need so little blood, but
Yeah.
we want to make sure the vets are fully involved initially to vet the technology and to counsel owners and stuff like this.
Yeah. Okay. Multiple cancer types. When they have multiple cancer, there's quite a few dogs who end up with several
Yeah,
different simultaneous
yeah.
cancer.
As you mentioned before, we just completed this test of 800 dogs in our trial, and so we're about 180 cancers that turned up in there in the control group. So there were a significant, and we did autopsies on a good share of these dogs that came through and were diagnosed. There were, even though they were diagnosed of having died of a specific cancer, let's say hemangiosarcoma, when the autopsy was done, a significant number, and I forgot that exact number now, actually had evidence of another cancer in addition to the one that was apparently caused the death. That this does frequently happen. Yes.
Okay. And I'm curious well, your test will catch that if hopefully, but it's
We
not going to
we
say,
we don't know that. That's actually interesting. We do know that we've had, like back to this study of pancreatitis and things like this, there were cases where people had heart disease and pancreatitis. We could distinguish those two from each other, but we don't know that. If somebody has a dog has hemangiosarcoma and osteosarcoma, will that be two separate signatures? Or so when somebody had pancreatic cancer and diabetes, they actually had a new signature that was different than both pancreatitis and diabetes. And they had this new one, but they still had the individual signatures. Whether that'll occur in dogs with two cancers, we don't know. We're hoping that will be less frequent if we're picking them up early. If we're picking them up at stage, more often the dogs that had autopsy had two tumors, they were discovered at a late stage. So we had a dog whose immune system was already being compromised by the initial tumor, presumably allowing for initiation of a secondary tumor.
okay. So any other questions, please put it in the chat. Let's see.
Okay.
Okay. Let me see if I have any that people have submitted when they signed up.
And if you think of something later, you can just email it. If you put in Calviri, our site will show up and you can email me directly.
Can you say just a little bit more about osteosarcoma? You had mentioned something about specificity. Yeah.
Yeah, we don't know exactly why, but it stands out as being the hardest one to distinguish from the other cancers. For most of the other cancers, we can say, okay, you are hemangiosarcoma, you are not these other 4 cancers. Osteosarcoma, that's harder for us to do. They look more like they might be one of the other cancers. We don't know exactly why.
Let's see. So I think how close are we so that once the test comes back and you're pretty sure that it's cancer, that you don't have to actually do conventional treatment, but just do immunotherapy and
Yeah.
caught it early, that you could actually get a cure with early
That's the ideal.
immunotherapy.
Yeah, no, you're exactly right. That's what we're aiming for, is that you don't go in and go get radiation surgery or chemo for your stage 1 tumor. You would get a, hopefully, maybe an off-the-shelf vaccine, which we know we can produce, but it may need to have some sort of adjuvant or so like a checkpoint inhibitor or something like this to boost that response. But that would be the ideal. You walk in, you don't, it comes back, the test says you have angiosarcoma, the vet pulls an off-the-shelf vaccine, gives it to you with gilvetmab or whatever, and that's it. That's all you have to do.
Right. But unfortunately, gilvetmab is like a $15,000 treatment.
Yeah.
So,
Unfortunately, that's the case. Well, which is a lot cheaper than Keytruda. But you're right. It's ironically, even in developing countries, they couldn't afford a $15,000 per human. So something's got to be done on that front in terms of what that adjuvant is. It can't cost $15,000.
right. Absolutely not. And these adjuvants are actually cheap. Many of them.
There are things out there. The adjuvant that we've used in this trial is called Hiltonol, and it's been used in dogs before, including direct injections into tumors with some mixed results. And it's basically double-stranded RNA.
Oh, okay. Good.
It's commercial. It's been in clinical trials. I think it's actually been approved for one kind of treatment for humans. So it's interesting. I will tell you this. We administered Hiltonol to dogs. It's been administered to people. I administered it to myself, had no bad reaction to it. But when we started our clinical trial for the preventative vaccine, a few small dogs had a very bad reaction to it. And so we had to cut the dose down in half from what we use in a mouse for the dogs because of their reaction, the smaller dogs we did it for all dogs, but because of this reaction, which is very peculiar to dogs.
Wow. Fascinating. Tell me about the regulatory approval process. So in human medicine, you have to go through FDA for any of these tests, but for veterinary world, tell me about the tests.
So this is part of the reason we chose to focus on the diagnostic first, is there's no regulation for dog diagnostics, unlike human. In human, you have to go through the FDA to get approval for your diagnostic. Not in dogs, and there's no prospect of the USDA requiring approval for that. We've asked them about it. For the vaccines, it's a different story. For biologics, like a vaccine, it's approved through the USDA. And in that process, you have to prove to the USDA that your product is safe and has some reasonable expectation of efficacy. And that can be based on mouse models or things like this. They will then give you what is called a conditional license. So you can start a clinical trial to get your product approved. But during that clinical trial, you can actually sell your product.
Once you have a conditional approval.
Yes, and which we plan to do. So we think our preventative vaccine may be available pretty quickly in the future by virtue of a conditional license. Now, you can't advertise it then. You can't give it a brand name and things like this, but you can start using it, and people can use it outside of the clinical trial. It can be, so that has an advantage. There's talk about doing that sort of thing on the human side also, but right now that's not approved.
I see. So how close are you to getting conditional approval for the preventative vaccine?
Yeah, that's going to be interesting. Right now, the limiting factor will be our discussions with the USDA, whether they can take the data we have on safety and efficacy and approve a different vaccine, because we don't want to use the same vaccine that we used before, because we know we can make one so much better. And if they say they will allow us to use that data to now use this improved vaccine, then we think we could get that conditional license early next year. If they make us go back and do another trial to validate the efficacy, then we'll be delayed by about a year.
A year, okay. Really, 1 year would be enough time for you to enroll aging dogs and share
Yeah,
it?
it's interesting. So we have a new clinical protocol now that's much improved from what we used before, where we can take our chip and screen dogs, even very old dogs, to make sure they don't already have cancer, and then monitor them on the chip on a regular basis, not wait till they get diagnosed by standard procedures. So we can reduce the time dramatically for running the trial and for even the cost for a trial from what we did before. So we think we can get the conditional trial done quickly.
That's fascinating. That's really fascinating. Tell me your thoughts about EGFR HER2 vaccine as a preventative vaccine.
Yeah, that's interesting. There's actually more interest. When we first proposed a preventative vaccine, everybody said, you're crazy, it won't work. I even have an editorial from Nature magazine telling me I should stop my research because everybody knows that it can't work, so it's raising false promises. The field was so against this. But just lately, there's more interest in a preventative vaccine. It's almost the world starting to turn. And one idea is exactly what you expressed, is to find enough common mutations that occur in tumors that you can make a preventative vaccine. And these are DNA mutations. The problems with those is that they're specific mutations. Most of them are not immunogenic, so that you have that problem. And even when they are, they have to match the HLA or the immunotype of the presentation that your immune system does for that individual. So you can have a specific mutation that one person or dog will respond to, but the other person or dog will not respond to at all immunologically. So when you add up all those limitations, the possibility of preventing from point mutations in the DNA is reduced. And whether it's commercially a viable product or not is debatable. BioNTech on the human side tried to do that, or KRAS actually, not EGFR, and find enough mutations that they could put together that this one occurs in 1%, this occurs in 2%. So they'll put them together, make a vaccine, give it to you as a therapeutic. They weren't trying to do it preventatively. They were doing it therapeutically with Keytruda. It didn't work. I don't hold a lot of hope for that strategy. And partly because I think our strategy of using these neoantigens from the RNA level, they are always immunogenic. Every one we've tested, because they're frame shifts, they're longer, they've all been immunogenic. So it erases that problem. And it also erases the HLA problem because there's multiple HLA sequences in each one of these frame shifts. So this may be getting too technical, but I don't think that strategy, I don't think personal cancer vaccines are going to be a good strategy. I don't think preventative personal cancer vaccines are going to be a strategy. So I think really the best thing in town is what we're doing. And the closest thing to that is Lynch syndrome, and there's 2 clinical trials going on right now. These are about 10% of the colon cancer patients have a genetic defect so that they create frame shift mutations from their DNA. And they're predictable because they're always in these microsatellites, the runs of A. So they're predictable. So they now have gone and say, this frame shift occurs in people with colon cancer from Lynch. We'll give it to them preventively. And those trials have just started. And so we'll see how that works. But if that works, then it really validates our approach very well.
I see. Well, that's very exciting. Okay, let's see. Clinical trial for preventive use. So Emily says, is EGFR HER2 vaccine going to be available in clinical trials for preventive use? Right now, there is no plan that I know of. They're working on just getting conditional approval as a therapeutic vaccine. Yeah. Okay, let's see. What is this one? I'm sorry. Here we go. Maybe Lena. Lena, you have a question? Maybe she's dropped out.
Oh, no, I just needed to take off mute. No question. I was just commenting that you had mentioned the price of Gilbert was like 15,000 or so, and I'm not sure if that has to do with each institution's pricing or markup, but I know that here at UF for a 4-dose protocol that we have for our melanoma trial, we have been able to, our cost has roughly been about 5 to 6,000. So we are trying to use it. Our cost of like carboplatin chemotherapy is around, it can vary. It can definitely be up to 10,000. And so in our next protocol, we plan to use Gilvetmab with OSA with our immunotherapy, but we're trying to price it as much as we can to be comparable to the cost of carboplatin, which is, yeah, closer to, for a larger dog, for a 6-dose protocol, probably be close to the $7,000 to $8,000 range. So I just was like curious about all the different price differences that might be out there in Gilvetmab and if that's going to be prohibitive of using it more actively in the clinical setting for other practices too. So I may ask our pharmacy how their pricing is a little bit better for that reason.
Yeah, that's interesting. You're right. There is a lot of price. I know the people at Merck, they ran into a lot of production problems initially, and so all those costs have to be embedded into the actual price of the product that they had out there. I'm not sure where it will settle. It also, at least on the human side, you have to maintain the dosage if you are getting a positive response. And so you don't know how long you'll have to continue this. And it is pretty clear that the T cells energize over about 1 month, at least in humans. So you have to keep giving that boost every month or so to keep the T cells on toes. So it's still I hope they can reduce it down to 5,000 or whatever, but it's still very expensive. That's still going to preclude most people from using it.
Yeah, there is another company called Betagenics. I'm not sure if you're familiar with. They have their own anti-PD-1 drug. I don't know that we've also used that in our trials. Obviously, it was experimental, so it was only for our mRNA protocols. And that company, I know they're in the works of trying to commercialize and make their anti-PD-1 commercially available. They're also working on a feline anti-PD-1.
Huh.
And I'm really excited to see what they do and how things move along for them. But of course, like getting things to market, especially when there's a competitor with it with Gilvetmab is very challenging. But they have been really great to work with. And they're definitely their costs because it's a smaller company and they're doing things on their own. I'm sure their costs may be high, but I think at some point when they can get to market, we may have another option. And their PD-1 drug quality wise and in our trials was comparable to get with Gilvetmab.
Oh, that's interesting. Yeah, I
Yeah.
didn't know that. Yeah, so Keytruda goes off, I think, in 29. So it'll be really interesting how that opens up in terms of basically biosimilars competitiveness, because I think technologically there are simpler ways to make a PD-1 inhibitor, a synthetic antibody, for instance. So you don't have to go through the manufacturing expenses that they did. And they've already plowed the ground, right? They've already shown it works. They've done it. They figured out the dosing. So I think, almost if you follow cases, the price will go down on these things. My hope is we don't even have to discuss those anyway, because we've got a vaccine that prevents tumor in the first place. So we don't have to talk about $10,000 therapies or not.
Yeah. So far, what we've seen is we think that the PD-1, anti-PD-1 we're still early. We have to produce more data here at UF. But so far, we do think the PD-1 may be making a difference in some of our combination immunotherapy protocols, but we still need to continue to enroll and we're trying to expand the trials even further. And we're using 2 different immunotherapies with it. So
Yeah.
we really felt like it has, we have seen some improvement in overall survival with our patients and time to metastasis. But again, yes, it would be amazing if our immunotherapies did it all, or our preventive cancer vaccines, because the immunotherapies themselves are, for the most part, easy to administer, don't have a lot of side effects.
Right.
And
you aren't putting, and they're not quite sometimes as invasive as even chemotherapy can be. And so, so that would be the dream one day.
Yeah, because even if that map's early, but if you look at the human experience, it still is very restrictive, the number of tumors that it actually can treat and the success rate for each one. So even in melanoma, which is their poster child, it's still only 30–40% of the patients respond. And they're not cured usually. It's a long-term treatment for these patients. But you are right, the adverse responses are much less to deal with than a lot of the chemotherapies that are out there, the same cancers.
So Elena works at University of Florida as a clinical trials manager. So she's on top of a lot of oncology.
Got it. I was picking that up already, got it down to go see who you are.
Yeah, I didn't quite introduce myself, but we had talked earlier in the week, and I'm going to start attending webinars and just trying to get more involved outside of my current role, just in the whole world of what's going on in oncology right now.
Yeah, good.
So here's a question. Pam is one of our volunteers asking, how far are we from a reliable canine cancer vaccine? And I think we'd like to know, Stephen, like for preventative vaccine to be reliable, what's the percentage? What's the difference in the sort of the number of, what does the difference in the curve look like? It's never 100%. So what is it that you're aiming for?
Yeah, so we're fairly, by modeling what we think the coverage will be, and if we can get good immune responses, the one thing, you have to get good immune responses clearly to get the protection, and it has to be a T cell response. Interestingly, for maybe Lana's interested in this, what correlates best with protection is CD4 double positive cells. So we have a good correlate immunity with protection, and we want to bring that in to the USDA as part of the trial. So we're hoping that we will get at least an 80% reduction in tumor incidence in dogs. We think that's an achievable goal.
That would be amazing. And once you have a conditional approval or a full approval, are you planning to track, or how are you planning to track the efficacy of the vaccine longer term?
That's a good question. I'm not sure because we don't have, in Australia, they have a registry for all dogs. You can get that data. As far as I can tell, that would be very difficult to do in the US, right? For outside of the trial, I'm assuming you're talking about how many of your dogs. Yeah, these are called like stage 4 studies, phase 4 studies in US in people. You'd have to afford those to do. They're not cheap to do that kind of follow-up. So that would be an open question. It's a good question, but an open question.
They're not cheap, and many companies are not really incentivized to do it if they already have original good data. But you want to get the real world data over longer term. So we're always curious, how can you design a study like that cost effective?
There might be okay, so this is getting into a little different area, but it turns out those same misprocessing that goes on in tumor cells also goes on in senescent cells. So it's a hallmark of a senescent cell is that they start misprocessing so they're making the same neoantigens same kind of neoantigens that tumors are. So we suspect that they may be some overlap in our cancer and senescent cells. And so there might be some effect on the incidence of other chronic diseases or the incidence of or how long dogs live as a byproduct of the cancer vaccine. So that would be a motivation to follow up on these dogs receiving the cancer vaccine to see what is happening to them in addition to the cancer.
In order to what? You're going to address longevity or?
Yeah. So many of you probably just saw the announcements about Loyal and what they're trying to do. And they just raised another $100 million for their C round. And so there's a lot of interest now in life extension and health extension in dogs. If I follow that area very closely, I think the most likely way to extend the life of a dog or make it healthier is to develop a vaccine against senescent cells.
Mm.
And I think our platform may be the best way to do that. We just don't have the bandwidth to start exploring that right now. But I think that's a good way to do that.
It's fascinating.
Senescent cells and tumors are very closely related. They're like the yin and yang in the same pairs.
Yeah. Okay. So if you could somehow tap into the community and have people voluntarily send you samples with necessary medical records so you can collect that data.
Yeah, it's an interesting, that's outside of my pay grade, how you would organize that. It would be a wonderful thing to do, but I don't know how to do it. Maybe do it in Australia, where they've already set up the medical record system.
If you find these educational webinars useful, and if you want to see more of these trials continue and new studies launch, please consider becoming a monthly donor. Thank you, everybody.
Thank you for watching. If you'd like to learn more about our work and available resources, please visit ccralliance.org. Together we can make a difference.